People from the back hear me?
Yes.
Okay. Okay. Thanks, Eddie, for inviting me to give a talk. It feels special to give a talk to colleagues here. So first, I want to acknowledge my lab, Erhu Cao, Cation-Chloride and has been drawing what I will present today in the past few years. And Sarah, a new graduate student who joined the lab recently, so she's also working on Cation-Chloride Cotransporters, mostly focusing on how these transporters are regulated by similar factors. Hopefully, I can talk about the first project sometime in the future. So we have been interested in sensory and the transport membrane proteins in the kidney since I started my lab here at the University of Utah. So the kidney filters about 180 liter plasma each day, but only produce 1 to 2 liter urines. So this, if you do the math, so you will realize the kidney has to resolve most nutrients, waters, and the salt from the filtrate back into the blood. And at the same time, has to remove metabolic waste and the drugs we take, the medications we take from the circulation. And to balance electrolytes, acid base, and also blood volume and blood pressure, the kidney has a robust sensory mechanism to detect perturbations of those key parameters, and then has a transport mechanism to restore homeostasis. And so you can think of kidney as a long-class sensory organ and has all those transporters, ion channels, and the receptors to sense changes in our body fluid and try to rectify perturbations. And such sensory transport mechanisms exist in other organs as well. But I think the most studied system, those systems is in the brain. For example, there are neurotransmitters that can activate voltage-gated ion channels to elicit electrical signaling. And also there are neurotransmitters that activate G protein coupled receptors to initiate the chemical signaling. And at the end, there are transporters can reuptake those neurotransmitters from the synaptic cleft back into the cell and permeate those events. And so we have quite a sophisticated understanding of those systems. For example, we know how like psychoactive drugs, antidepressant and the cocaine acts on these ion channels, transporters, and how those drugs influence our mood, emotional state. But I think in the kidney, we don't have such nuanced understanding of those transport and sensory mechanisms. So that's what my lab trying to understand using mostly structural biology approach. And so when I started my lab at the Utah, so we started working on polycystic kidney disease protein, PKD1 and PKD2. Those are proteins, those are genes that mutated in autosomal dominant polycystic kidney disease. And so we are working on structure and function of polycystic kidney protein in the lab. And more recently, we also worked on Cation-Chloride Cotransporters. So that's what I will tell you guys today. So I would say there are three major discoveries that have shaped research of Cation-Chloride cotransporters. The first one is the gas discovery of diuretic drugs in between 1950s and the 1970s. So it started with this compound called sulfur linamide. So clinician found in 1950s that this compound, which was used as antibiotics at the time, has some diuretic effects in patients. Then the company started getting excited of this compound because at that time only mercury is commonly used to treat edema, fluid overload condition. But you can see mercury is very toxic compound. It's not ideal. So the company started to test derivatives of sulfur-linamide, and eventually they found three different classes of compounds, derivatives of this parental molecule that can function as a better diuretics for the treatment of edema and also hypertension. So one class is called carbonic anhydrous inhibitor. So these compounds act at the proximal tubule. And another class of compounds is called cyanide diuretics. And the people later found out the cyanide diuretics inhibit sodium chloride reabsorption at the distal convoluted tubule. And a third class is called loop diuretics. And so from the name, you can see that these drugs inhibit ion reabsorption at the loop of Henle, ascending limb of loop of Henle. So the two commonly used loop diuretics are furosemide, bumetanide, another one is called furosemide. And so one thing I want to point out is that so all these compounds inherited this so-called sulfomoid group from the parental molecule. And so you cannot give patients these drugs when they have sulfur energy. So there are probably approximately 7 to 8% of patients cannot take this type of drugs because of sulfur allergy. So one goal would be to come up with compounds doesn't have sulfur and can be given to those patients. So the second major discovery, I would say, is the cloning of Cation-Chloride cotransporters as the targets, or as the molecular targets of loop and the cyanide diuretics. So now we know there are 7 members, 7 Cation-Chloride cotransporters in human genome, and they can be divided into two groups. So there are 4 potassium chloride cotransporters, and these transporters are sodium independent. And there are 3 sodium dependent cotransporters, Cation-Chloride cotransporters. So NKCC1, NKCC2, so co-transport 1 sodium, 1 potassium, and 2 chloride, and sodium chloride cotransporter transports 1 sodium and 1 chloride in each transporter cycle. And all these transporters are electroneutral, means they don't generate membrane potential during their transport cycle because the negative charge and the positive charge are balanced during the transport. And also these transporters are secondary active transporters, meaning that they don't have ATPase domain to power their transport. They rely on the ion gradient generated by sodium potassium ATPase, sodium potassium pump to drive their transport. For example, NKCC, NKCC1, NKCC2, and the NCC use the inward directed sodium gradient to transport the sodium, chloride, and potassium. They are called like ion loaders. And the potassium chloride transporters, KCC transporters, use the outward directed potassium gradient to introduce potassium chloride from the cell. They are called ion extruders.
Just a quick question before we get into some additional detail. So with the with these transporters, the SGLT2 inhibitors, is widely used in this context. But I'm just curious to know if there's any sort of interaction between these inhibitors and this sort of cascade channel for transporters.
Yes. So I will discuss here. So the third major breakthrough in cation-chloride cotransporter research is the human genetic study pioneered by Richard Lipton group. And they found that mutations in NCC cause a genetic disorder called Kidman syndrome, and the mutation in NKCC2 underlie another genetic syndrome called Barber syndrome type 1. So basically, this genetic study highlights the pacemaking role of NKCC2 and NCC in renal salt and water reabsorption. And those two diseases are characterized by, because those are loss of function mutations, and those patients have low blood pressure, and they have like also sort of wasting a phenotype. They kind of like sort of craving. They need to take more salts to balance their electrolytes. And they have no blood pressure as it's common for those patients to have no blood pressure as well. Okay, so I also wanted to say that NCC reabsorbs about 7% of the salt at the distal column to the tubule, and the NK62 reabsorbs about 30% of the salt at the ascending limb of loop of Henle. And so a lot of breakthrough in the research also pioneered by Lipton group is that they found that mutations of kinases, Wnt kinases, and their upstream E3 ubiquitin-like degrader cause a larger mirror symptoms. It's called Gordon syndrome. Those are hypertensive syndrome. And then people later understand that there's a kinase pathway that regulates CCC transporters. So for example, Wnt spark kinases activates NKCC1 through phosphorylation. But at the same time, this kinase pathway inhibits KCC. So KCC and the NKCC are inversely regulated by phosphorylation. So phosphatase has the opposite effect on NKCC and KCC. And go back to your question. So the thiazide diuretics inhibits NCC and basically just block ion reabsorption at the distal convoluted tubule. And the loop diuretics inhibits NKCC2 at the ascending loop of Henle, and they are both used for treating hypertension and fluid overload condition caused by liver disease or congestive heart disease. So let's start with loop diuretics. So we know loop diuretics inhibits NK62 to exert the therapeutic effect, but also we know the loop diuretics also inhibits NKCC1 almost at equal potency, and at higher potency, loop diuretics also inhibits KCC transporters, and so those loop diuretics are used widely in clinic. There's more than 30 million prescriptions each year in the United States, but they are not perfect. So the off-target inhibition of NKCC1. So in the next two slides, I will explain to you that off-target inhibition of NKCC1 and the KCC can cause the side effect of hearing loss. It can be permanent, and in most patients, it's temporary. And also, I will explain to you that inhibition of NKCC1 can be used, can be harnessed to treat brain disorder as well. So the pharmacology of loop diuretics is a little bit complicated. It has side effect, has therapeutic effect, but the side effect can be harnessed to treat brain disorder. So let me explain to you. So why loop diuretics can cause hearing loss in some patients? So this is because NKCC1 and the KCC3 and the KCC4 play a very important role in secreting potassium into the endolymph. So endolymph is a kind of a typical extracellular ionic compartment. So in normal cell, the extracellular fluid has high concentration of sodium and low concentration of potassium. But in the endolymph, it's kind of strange. It has very high potassium concentration and very low sodium concentration. And this compartment is achieved through combined action of transporters and ion channels. For example, in supporting cell, so the basal-lateral NKCC1 and the potassium ATPase, sodium potassium ATPase mediates like a potassium entry from the basal-lateral membrane side, and then the apical potassium channel mediates secretion of potassium into the endolymph. This is a combined action of ion channels and the transporters, and similarly, KCC3, KCC4 expression in other supporting cells has the similar function, just recycle potassium into the endolymph, and the potassium ion is then used by mechanosensitive ion channel expressed in the hair cell in response to wave stimuli, like sound wave stimuli, and these are like potassium selective ion channels. So they need a high extracellular potassium to function to initiate mechanical construction. So then it's not surprising NKCC1, KCC3, and the KCC4 not all the mice are profoundly dead, and loop diuretics can cause hearing loss in some patients because inhibiting NKCC1 and to a lesser extent KCC3 and KCC4. Okay, so I mentioned that inhibiting NKCC1 can be beneficial in treating brain disorders. So this is because NKCC1 together with KCC are important in maintaining intracellular chloride concentration in the brain, in the neurons of brain. So in mature neuron, normally the NKCC1 activity is high, and there's almost no expression of KCC. Then in this immature neuron, the chloride concentration is high, and then the GABA, which is a classic inhibitor neurotransmitters, can elicit chloride efflux because the intracellular chloride is high. The chloride go out of the cell, then this actually depolarize the neuron, and this excitation of immature neuron. This is very important for developing brain because they need those exciter action for circuit wiring and the formation of the developmental stage. But once the neuron immature, the NKCC1 expression is low, and the KCC expression is high. So you can see that the KCC mediated chloride extrusion will maintain a low chloride concentration in immature neurons. Then the GABA neurotransmitter can inhibit excitation of the neuron because the current will be outside and the chloride flow will be inside. So this model actually is also supported by human genetic studies. So mutation in KCC in human can cause epilepsy and seizure, and the mutation in KCC3 is associated with peripheral neuropathy. And also many brain disorders often has imbalanced excitatory versus inhibitory synaptic transmission. So then you can imagine NKCC1 antagonists and the KCC activators can be used to restore GABA inhibition, and there are potential candidates for treating brain disorders. So actually, loop diuretics has been repurposed in some clinical trials to treat brain disorders, but with like non-convincing results, mostly because loop diuretics has very low probability to cross the brain-blood barrier because they have like charged groups, which I will show later.
Can I ask a question? So I was wondering, the previous slide, where you had the previous slide, I'm not sure, where you had the activators and the inhibitors. I guess what I'm thinking is it possible under physiological conditions to have multiple combinations of this subunits in assembled channel complexes?
Say it again.
Okay, so with can we go to the previous slide? I think might be easier if people look at the slide. Yeah. So what I'm thinking, yeah, it was just the one before that one. But anyway, what I was just thinking is that is it possible? You talked about the activators as well as inhibitors, right? Mm-hmm. Maybe relevant, but what I'm thinking is it possible that in assembled channel complexes or cotransporters, could you have this multiple combination of subunits?
Has multiple
combination of this different channel subunits. I don't know if that makes any sense or not.
So you wanted to have like multiple?
Yes, that's what I mean.
Drugs?
Yeah.
Drugs on the cotransporter?
Right, because you could have the different, subunits together in assembled the subunits. Oh, we can talk later.
Okay.
Okay. Yeah. All right, sorry about that. Yeah.
Okay, so this loop diuretics and thiazide diuretics has been extremely successful in clinic. There's probably like more than 30 million prescriptions each year. So it clearly show that the CCC transporters are druggable. But the problem with these drugs, so I mentioned the one, the suffer energy, and hearing loss, those kind of side effects. And also, so in the lab, we try to understand how loop diuretics and cyanide diuretics works. Then hopefully we can learn from that and try to come up with better drugs to target NKCC1 or NKCC2 specifically. So now the loop diuretic cannot differentiate these two transporters. And so we can also think about how to remove the sulfur from those compounds. And so those will be useful for treating patients with sulfur allergy. And another fundamental aspects of CCC biology, I mentioned a couple slides earlier, is the phosphorregulation. So from the human genetics, we know the kinase mutation can cause hypertension. So that phosphorregulation is fundamentally important for CCC. But we don't have a mechanistic understanding of how phosphorregulation works. And so the next question is, if we learn how phosphorregulation works, can we try to target that phosphorregulatory pathway to come up with different types of diuretic drugs to treat hypertension and fluid overload? So this is a KCC1 structure. So I wanted to use this structure to show some key structure features of CCC transporters and their ion binding sites. So CCC is a member of a larger family of transporters called amino acid polyamine organocation transporters. So similar to those transporters, which we have structured before CCC transporters. And so you can see here is that the TM1, which is shown in blue, and TM6, which is shown in green, they kind of unwind at the middle of the lipid binder. And this unwind region turns out to be very important for binding ions. So you can see two chloride ions shown in green, and the one potassium ion shown in purple, coordinated around that like discontinuous helical region. And so another thing I want to point out is that this chloride just sit above the potassium, and they actually interact with each other. They are like co-transported through the transporter cycle. And this NKCC1 and NKCC2 transporter has an additional sodium site, also near also around the discontinuous helical region. And NCC doesn't have the potassium binding site. Instead, it has a sodium binding site. So that's the only difference. And structurally, all the CCC transporters are very similar. Okay, so this is a structure we determined of NKCC1 bound with dimethylamine. And the third most prescribed over there is in the United States. So what you can see here is that there are two things. So first, so the dimethylamine arrests the NKCC1 transporter in an outward open state. So when the transporter transport ion, they oscillate between three fundamental states. So the ion binding site can face to the extracellular space. It's called outward open state. Then it cools off and transition into an occluded state. In that state, the ion binding site are inaccessible from both extracellular side and the intracellular side. Then it opens to the inside, then I can escape into the cell. So the dimethylamine captures the transporter in an outward open state. And the second thing I want to emphasize is that Pimetanide co-occurs one potassium ion, one chloride ion, and one sodium ion. And the chloride ion just above the potassium is replaced by the carboxyl group of pimetanide. So the carboxyl group of pimetanide now interact with the potassium ion. So in the slide before, I showed that chloride and the potassium sit close to each other and they interact with each other. So basically, in this case, the pimetanide kicks out one chloride ion and then interact with the potassium ion. And so this actually explains a lot of all the literatures. We didn't pay attention at the beginning. So in 1980s, people noticed that pimetanide binding, radio-labeled pimetanide binding requires all three ions. And the chloride ion has a peculiar effect, has a so-called biphasic effect. So up to 5 micromolar, it stimulates pimetanide binding. Then when the chloride ion increase further, then it starts to inhibit the pimetanide binding. And the potassium and the sodium does not have that effect. And so this makes sense, right? Because the pimetanide kicks out one chloride and keeps one chloride. So that explains the biphasic effect. So the buffer we use to determine this structure has 5 millimolar chloride. That's the key. So we start with normal sodium chloride buffer, and we never get it work. So when we realized that this all the literature, then we started to play around with the sodium with ionic condition, and then we got this structure immediately. Okay, so later we also have a NKCC1 structure bound with furosemide and also bound with torsemide. So the common thing is that furosemide and torsemide also bind approximately the same pocket as does pimetanide. And so one important difference is that the torsemide slightly reaches deeper into the ion translocation pathway, and it actually encroaches the potassium side and expels the potassium from this side. So this one is a water, is not potassium. And in the case of furosemide, the potassium is also important for binding. So I will explain in more detail in this slide. So these are the binding side for the 3 different leucodiuretics. So you can see here, furosemide and the pimetanide all contain a carboxyl group. And this carboxyl group interacts with the potassium ion. So these 2 drugs, these carboxy-containing leucodiuretics, can be called potassium-dependent inhibitors of NKCC. And you can also see the sulfonyl group of furosemide and pimetanide interact with this arginine. These arginine are important for opening and close of the extracellular gate of the transporter. And in the case of torsemide, there's no carboxyl group there. It lacks the carboxyl group, and it reaches deeper and kicks out the water ion. And the sulfonyl group is also here in the torsemide, but it still doesn't make many contacts with the transporter. So this led us to classify the leucodiuretic into 2 groups. So it's one is potassium-dependent antagonism, and another one is potassium-competitive antagonism. So we did one experiment and show that in the case of furosemide, if exclude potassium, it actually has the protein has less potent, become less potent without potassium. Okay, so we also have a structure of NKCC2 found with bumetanide. NKCC2, remember, is the therapeutic target of leucodiuretics. And people has noticed for many decades that leucodiuretics cannot distinguish NKCC1 and NKCC2. They inhibit NKCC1 and NKCC2 pretty much at equal potency. So when we have the structure of NKCC2 bound with bumetanide and compared with NKCC1 bumetanide structure, it's clear to us that these two structures almost superimposable. There's almost no difference there. And the ion translocation pathway and the bumetanide binding pocket are almost identical in these two transporters. And so I think if we want to develop an isoform specific leucodiuretics, we have to target other area of the transporter, not the ion translocation pathway, because they are so identical. So now let's move on to talk about thiazide diuretics. So thiazide diuretics can be divided into two groups. So the one group is called classic thiazides, and because this class has the benzoyl-thiazine ring here, and another group is called thiazide-like, and they don't have that classic ring, benzoyl-thiazine ring, and nonetheless, all these thiazide type diuretics has this submolar group, and they also have this chlorine atoms in their structure. So this will become important later in my talk. And so these are the three structures we determined using human NCC transporter, and together with the three most popular thiazide drugs used in the United States. So hydrochlorothiazide is the classic thiazide drug, and the indapamide and the clozapine are the thiazide-like drugs. So what you can see from here, so these three drugs has a similar binding pockets, and they have pretty much similar inhibitor mechanism for blocking ion reabsorption. So now let's compare leucodiuretics with thiazide diuretics. So I included several residues as a reference for comparison. For example, this arginine 307 in NKCC1 is the gate residue, is actually extracellular grade residue in NKCC1. The equivalent residue in NCC is arginine 157. They are at the same place, at the same level for comparison. And this tyrosine in NKCC1 is important for coordinating potassium. And the equivalent residue in NCC is this histidine. And this histidine is important for coordinating sodium. So they are at the same level as well. So from here, you can clearly see that the binding site for leucodiuretics is more superficial. It's more close to the extracellular space. And the thiazide drugs reach much deeper into the ion translocation pathway. So in the case of bumetanide, the potassium ion is remained in the structure and is important for binding. And the chloride ion is also co-occluded for binding. And in the case of thiazide drugs, you can see that chlorine ion is just gone. This is because I told you earlier that all the thiazide drugs has one chlorine atoms in their structure, and that chlorine atom actually clashes with the chlorine side. So it's kind of a chlorine atom replaces the chlorine ion. So to put it simply. So the second thing we noticed is that, so remember the loop diuretics and thiazide diuretics inherited that submolar group from the kernel molecule, but they are used differently. So for example, in the loop diuretics, that submolar group is face up, and in the thiazide type drugs, that submolar groups face down. So they have different poles when the inhibitor interact with their targets. Okay, so the one more drug bond structure. So this is a structure of KCC1 bound with a inhibitor. It's called a VU0463271. This is a tool compound. It's not a clinical drug. So when we determine that this structure, it's clear to us that this inhibitor also targets the ion entryway of KCC transporters. It's kind of has a similar mechanism as loop diuretic and the thiazide diuretics. They all targets the extracellular ion translocation pathway. And in this case, this drug is potassium competitive. And in the structure, we can see this drug reaches deep into the ion binding site and clashes with the potassium binding site. So all together, it shows us that the extracellular ion translocation path is a hard spot for pharmacological inhibition of CCC. So all these different chemicals ended up targeting the same analogous site in their targets, in their CCC targets. Okay, so we determined the structure potassium-free buffer. So in this case, we know the potassium inhibitor potency. Okay, so now let's talk about the phosphorregulation of CCC transporters. So we are motivated to study phosphorregulation of CCC because this is a fundamental regulatory mechanism for CCC transporters, validated by human genetic studies and also by a lot of biochemical studies. So the rationale is that maybe the phosphorregulatory pathway more different, more divergent in CCC than the ion translocation pathway. Then it may offer a better opportunity to target phosphorregulatory process to achieve isoform specificity. So that was the rationale. So before we started the project, we can think of two mechanisms how this phosphorregulation work. It could be just phosphorylation brings the trafficking of these transporters to the plasma membrane more readily, and or maybe their degradation is suppressed or something like that. And a second hypothesis is that maybe phosphorylation change the structure of the transporters and then change the ion transport rate that way. So it's a more direct mechanism. So we actually favor the second mechanism. So that's why even the first mechanism, you never see it in structure. So we just assume it may be the second mechanism. So we'll try to determine a phosphorylated CC structure try to see the impact conformational change triggered by phosphorylation. Okay, so the phosphorylation has been shown to regulate membrane protein in a lot of cases, but it's kind of difficult to study using structural approach. And the main reason for this is that how can you purify an authentically phosphorylated protein and to do a structure with that phosphorylated protein? So we have tried many times, and eventually it worked when we co-express either NCC, NKCC1, or NKCC2 together with the upstream kinases. So in this case, we co-express WNK1, which is we use a constitutively active WNK1. We know that WNK1 is active in our system. Then we also co-express WNK1 downstream substrate called SPA kinase. And there's another protein called MO25, which is a scaffolding protein that stimulates kinase activity. So when you express WNK, SPA, MO25, we also express YMP, which is a marker, fluorescence marker for stable sunlight selection. So once we have this, and we then show that the transporter is indeed phosphorylated. So in the case of NKCC1, we did the mass spec. We can clearly see phosphorylated serine in the sample. In the case of NCC, we use a functional antibody, and we can see that the protein we purified is also phosphorylated. And so we and others have determined inactive non-phosphorylated NKCC1 structure many years ago. So in these structures, the cytosolic domain is either invisible, is not defined in the structure, or it loosely interact with the transmembrane domain. So there's a big gap between the cytosolic domain and the transmembrane domain. And this means that without phosphorylation, the cytosolic domain are kind of flexible, has different orientation with respect to the transmembrane domain. So that was what we learned using the non-phosphorylated sample. Okay, so these are the phosphorylated CCC transporters. So this is NKC phosphorylated NKCC1 bound with rosemide. This is phosphorylated NKCC2 bound with juvenilide. And this is phosphorylated NCC bound with indapamide. So we can clearly see the phosphorylated serine or threonine residue, so which are shown in orange here. The phosphate group are shown in orange here. So you can see NKCC1 and NCC has three phosphorylation sites that's clearly visible in the map. And in the case of NKCC2, there are 4 phosphorylation sites, has 1 additional phosphorylation site. So I will come back to that later. So what does the phosphorylation do in all these structures is that phosphate is negatively charged. So once these residues get phosphorylated, they become negatively charged. Then they start to interact with positively charged residue, such as adenine and lysine. And so this interaction, phosphorylation diffuse interaction, foster formation of 2 domain interfaces. So one couples the N-terminal region with the cytosolic domain, with the C-terminal domain, which is here. So this phosphorregulatory segment is invisible without the phosphorylation. It just flopping around and you never see it. So now once phosphorylated, it binds to the C-terminal domain of the transporter. And then once this N-terminal domain, C-terminal domain complex form, to start to interact with the transmembrane domain. So there's a charge interaction here. So in NKC1, there's arginine at the bottom of transmembrane domain interact with this phosphorylated serine. And in the case of NCC, it's the two nitrogen residues interact with the phosphorylated serine. And in the case of NKC2, it's arginine, again, interact with phosphorylated serine. Okay, so what we think now is that the phosphorylation leads to formation of these two dual domain interfaces and stabilize the transporter in a conformation that is conducive to rapid ion translocation. The transporter can breathe more quickly than non-phosphorylated transporter. So, okay, so why NKCC2 has one more phosphorylating site? So this is kind of strange, intriguing to us. So this is the alignment of phosphorregulatory segment in NKCC2, NCC, and NKCC1. So I showed you NCC and NKCC1 use the site 1, site 2, and the site 3 to promote the domain interaction once phosphorylation. And the site 3 is also in NKCC2. It's also phosphorylated, but actually it's not used for interaction with the transmembrane domain. But site 4 in NKCC2 is used to interact with the transmembrane domain. And so this is kind of surprising to us. And so we know for decades that all mammalian NKCC2 has 3 splicing isoforms. And they differ in the region that precisely interact with the cytosolic domain. So remember, this arginine 240 will interact with this phosphorylated serine. And the 2 residues before this arginine are different in 3 isoforms. So it can be cysteine in B, phenylalanine in A, and the valine in F. So that made us to think that maybe the different isoforms has different coupling strengths between the transmembrane domain and the cytosolic domain upon phosphorylation. And these 3 isoforms has different physiological role in the kidney, and they have different ion transport properties. So the current project in the lab is trying to understand whether phosphorylation difference in 3 different isoforms underlie their different ion transport. Okay? And there are conclusions on I just list here. So hopefully these structures can help us to start to think about design better drugs for treating hypertension and overload. And finally, I want to thank people who helped us. And thanks for listening.
Questions?
Define. Define.
Great talk here. Question. Given that the cavities that these girls want,
You have to speak louder.
yeah, 2 questions. One is what, so if the